Hi, I'm Dr. Marla Hilderbrand-Chae. You might remember me from such science educational videos as the Algae Scale-Up project. And that number that you get, multiply. And the summer learning.
Coming out of someone else.
No, it's just like
Today, I'm going to talk to you about how this little hydrogel can revolutionize gene therapy. While I am unlikely to have that level of impact on gene therapy here during my PhD, I am excited to talk with you today about my project involving polyacrylamide hydrogels. Polyacrylamide hydrogels can be made by combining chemicals and forming a matrix. And generally in cell culture, we do these on glass cover slips. Various extracellular matrix proteins can be added to these hydrogels in order to create different cellular environments representing where they grow in vivo. The cover slips are then put in plates and cells are added, media, and the cells begin to grow. The really wonderful quality about polyacrylamide hydrogels are the ways in which we can tune their properties to create different living environments for cells to grow on. This is an example of how we can pattern polyacrylamide hydrogels in order to create different cell shapes, resulting in different epigenetic landscapes. We can also vary the stiffness of the hydrogel and have an effect on how it grows in culture. You can see in this figure the increasing levels of stiffness and the morphology of the cell as they're grown at these levels of stiffness compared with how a cell would grow on tissue culture plastic. Some really exciting early research from Dr. Yanfen shows that the cells grown on polyacrylamide hydrogels can be more likely to be found in S phase of the cell cycle compared with the tissue culture control. This is very exciting because this means that just growing these cells on the hydrogels can give us some advantageous properties for gene transfer. There are viruses that are more likely to infect a cell when the cell is in an S phase. And the question becomes, can we get this higher percentage of gene transfer? These are my references, and if you want to hear more about my amazing project, vote me through to the next level.